So, how a T cell interact and activate B cell? As we know about APCs are the antigen presenting cells, and they consist of B cells, dendritic cells, Langerhans cells, and macrophages. All are APC cells. So first, the APCs present cells with T cells, so they are antigen presenting cells, and also there is a rule number 1 is two signals are compulsory for T cell activation, B cell activation, and class switching. Okay, so signal 1. First, signal 1 is activation of T cell activation, and signal 2 is activation, and signal 2 is T cell, and signal 2 is proliferation and survival. So this is a dendritic cell. We know that dendritic cell, they are the antigen presenting cell. So they have antigen, they come to the dendritic cell, and then dendritic cell differentiate this antigen into either it is an exogenous or it's an endogenous. If it's an exogenous, then the dendritic cell express a receptor, MHC receptor, with the antigen. If it is an exogenous, then MHC 2, and if the antigen is endogenous, then the receptor is MHC 1. So this is the MHC receptor major histocompatibility complex. Antigen came, the APC process it, recognize it, what the type of antigen it is, then they express its receptor on, then express the antigen to the MHC receptor. Then this MHC receptor is further it goes to the lymph node, where the T cells in the lymph node, where the mature T cells are present, and these mature T cells, this is the mature T cell. The T cells have TCR. This is the TCR, and this TCR signals CD3 for signal transduction. So this CDR, it is CD3 is for signal transduction. Okay, then we have this TCR is we can say it's we have read that the TCRs bind to the specific cell surface receptor, cell surface proteins like CD4. So if the MHC2 is expressed or it's bind to TCR, then the TCR transduced and release CD4 receptor, CD4, and if the TCR recognize an MHC, then it expressed CD8 cell. This is a knife. So this is called the T cell activation, in which we just give antigens from the APCs to the T cells. The next step is T cell proliferation, and in this process the dendritic cell also expressed this is signal 1, Now signal 2 is the dendritic cell expressed another uh, another receptor which is known as B7 or CD80 or 86 B cell receptor, and it has a receptor on the T cell which is known as CD28. So the interaction of B7 and CD28 can cause proliferation and survival and activation or release of cytokines. So T cell activation is processed in these two signaling pathways. First pathway is activation of the T cell, and second pathway is proliferation of the survival. So this is all the APC, how a dendritic cell activate a T cell. Now we can we will learn another APC example like B cell. Now we know how a B cell activate a T cell. So in B cell, we know that the antigen is endocytosed into the B cell. So it's an endocytosis process, endocytosed. So it's an exogenous, and this is exogenous antigen because it is endocyte into the endosome. Now this leads to the expression of MHC2, and the MHC is expressed on the antigen is expressed on MHC2 receptor of the B cell, which again activate the TCR of the T cell, which is actually activated T cell. The TCR recognize release CD3, which activate or which gives the CD4 surface protein. So this CD4 surface protein, now the antigen first the activated T cell, it's activated, it means that the CD4 is already expressed. So whenever the the exogenous antigen, B cell present an antigen, B cell always present antigen which are exocytosed. This leads to the expression of the MHC2. This MHC2 goes present this antigen to the CD4, and then this CD4 it releases CD4. So this is a signal 1. Signal 1 and 2. This is actually the T cell activation. Now in B cell activation, or B cell activation first, T cell activation. This is the process of T cell activation or signal 1, and now signal 2. In signal 2, B cell release express another cell surface marker which is CD40, and its complementary receptor is CD40 ligand, and it releases ligand. It activates it also proliferate and survive and release cytokines. These cytokines are released from the T cell, and they determine the activated B cell class switching of immunoglobulin. They determine which immunoglobulin is class switched and released into the bloodstream. So this is all about how a dendritic cell, how a B cell is activate a T cell. There is another a small pathway. Macrophages also activate T cell, and in this pathway, macrophages expressed another CD40, ED40 and CD40L. So this activated first the T cell releases interferon gamma, and this goes to the macrophages. In the macrophages, they release Janus STAT kinase pathway. STAT activates the Janus STAT pathway, activates the STAT translocation, and this goes to the nucleus, releases interleukin 2, in which releases interleukin 12, and these interleukin 12 goes to the interleukin 12 receptor at the T cells, which again enhance the production of interleukin. It also releases cytotoxic enzymes to kill intracellular bacteria in the macrophages. So in macrophages we have CD40, which binds to the CD40 ligand, and interleukin 12, which is expressed, which goes to the interleukin 12 receptor of T cell, which activate interferon gamma. Intracellular bacteria come. This is like endogenous antigen. Intracellular bacteria like TB, and they are actually intracellular bacteria. They follow the pathway of MHC2 previously discussed. So they releases they goes intracellular bacteria like TB. They come to the CD14 or toll-like receptor, the innate immunity activated, come into the cell, and this releases and this activates macrophages and present this signaling intracellular pathogen signaling to the T cell, and this T cell releases interferon gamma, which activates Janus STAT kinase pathway, and they try to kill intracellular bacteria, and if they fail to kill, then they can wall off this intracellular bacteria forming a granuloma. So interleukin 12, if this receptor is defective, as we have discussed in T cell receptor diseases or immunodeficiency, then the body cannot releases interferon gamma, and if it not release interferon gamma, then it needs interferon gamma in order to cope up with this granuloma condition. So this is all about how a T cell, how a B cell, how a dendritic cell, how a macrophages are activated